today class we will be talking about the isolation of the protein. Now the isolation of protein, specifically we will talk about the yeast. Now if you look into the yeast, which is one of a very popular model organism, like the first eukaryotes whose genome has been sequenced in 1996 because of genetic amenability, short generation time. It is preferred as a model organism. Several Nobel Prizes for its cell cycle discoveries is also been studied in yeast. So they possesses a 23% homologous genes to humans. Several studies have mentioned that yeast can be used as a model organisms for Alzheimer's disease. In fact, in some of the cases, they have been used for cancer model or to study the apoptosis. Now, if you look into the genome, it is roughly like 12 MB. Okay, so that is a 12 MB genome size, 16 chromosomes in haploid. Again, one of the beauty with yeast is, and I'm specifically talking about Saccharomyces cerevisiae, the budding yeast or the baker's yeast, that they have a haploid and as well as a diploid life cycle. So that makes it more amenable, because if you disrupt a gene, then you can very easily work upon 16 chromosomes, roughly 6,000 genes which is present. Some places you refer 6,200. Now if you look, because our idea here is the protein part, so if you took into the protein total amount which is present, this is roughly in the femtograms, because we are talking about a single cell, by the way. Now if you look into the number of the molecules, okay, so in one estimation there are like 10 to the power 6 5 newtons per 6 protein molecules per cell. Okay, and this is based on the assumptions that more or less the protein size is 50 kilodalton. By the way, the protein size differ. I will show you a 2D gel picture as well to make you understand. So this is about the yeast and the protein content about the yeast. And if you look into the isoelectric points, because this isoelectric point is become very important for 2D gel electrophoresis, where you have to see the proteins bands, or also importance for one of the purification steps in terms of ion exchange chromatography. The masses also becomes important. That's why I'm mentioning here, because this masses is one of the gel or the size exclusion chromatography which is used to purify the or to separate the proteins, is also take into consideration of the size of the protein into consideration. So I will be talking about this in a moment. This is just a brief idea. Now if you look into a yeast cell, now the yeast cells is consist of a cell wall. Okay, and there is the central part, that is the nucleus, vacuoles. There are bird scars, and these bird scars, in fact in some of the pictures, it will appear like this type of yeast they shows. Okay, now these bird scars, which if you go into electron microscopy you will find like this also tells you about the age of the yeast cells. Very easily you can find out that how many budding this particular yeast has gone through. So that is a bird scar. Now the idea here is we are interested in knowing, or you are isolating all the proteins inside a yeast, or maybe you are also using a yeast as a heterologous expression system. In fact, like some of the yeast like Pischia pastoris, which is industrially important yeast, again similar to, you can say, the Saccharomyces cerevisiae, is used for extracellular protein expression. Means you can express a like human insulin gene is already been expressed in Saccharomyces cerevisiae, and you will find it into the market. So that's why one is interested in knowing that how we can purify this protein. The cloning part is something different, which can be dealt in some different lecture, but we are specifically talking about how we can isolate the yeast and what are the basic procedure. The size of the yeast, if you look into a microscope, then it will appear like around 5 micrometer. So that's the size. Okay, and they were oval in shape. Now, to coming back to the protocol part of the yeast, the yeast essentially consists of the same which we discussed in our earlier lectures also, the homogenization. The homogenization is like the terminology also used for mammalian cell, but it is the cell lysis which you are doing here. Then you are using a centrifugation, that is applying a centrifugal force, and the third after that you are basically doing a separation technique. So we already dealt with this thing, but I will also tell you the composition maybe in part 2 of this video, where you will look into what's the buffer composition is used. But essentially in today we will be talking about the centrifugation steps and the separation technique. So focus on that. Centrifugation, most probably in the previous lecture we talk about that, how what is RCA, what is G value, what is the RPM values as well, and the differential centrifugation for the isolation of mitochondria was explained. Now we will focus here in the separation technique. In the separation technique, the first things which you take into consideration, the property, that is the solubility, and one of the property which is called as salting out. I will be talking that in a minute. The second one is based on the size, which I was referring to the size in the beginning of the protein. The solubility of the protein is also associated with the charges in the protein as well, because we know that protein has got the amino and the carboxylic group. So they do also like positively charged proteins, there are negatively charged proteins, we know. So that's taken into consideration. So we'll see that in a moment. The gel filtration which I was referring, I will talk about that. Then what is the process of the dialysis, then the charge which is also employed for the ion exchange chromatography, where you use a cation exchanger or whether you use a anion exchanger. So briefly I will talk about this. And the third one, which is basically your affinity chromatography. Now this affinity chromatography is like for the neutral proteins or the amino acids which is present, and it's basically the idea is the antibody antigen reactions you were preferentially wants to uh, elute out or separate out the proteins of your interest from the rest of the other proteins, because the impurities which you are getting during the preparation of the protein is not like you are getting the DNA or the RNA and the cell debris, but sometimes you are interested in one particular protein because you are working on a recombinant DNA technology. Now there are several molecules, as usual, millions of molecules of proteins in a particular cell types. How to separate out your own protein? So people use this different techniques like affinity. Again, one thing which is will be dealt later on, that is the SDS page which is used, that is sodium dodecyl sulfase polyacrylamide gel electrophoresis. A separate video will be dealt with that. Now let's look into this. What is the mechanism of the salting out? Now in the salting out, is this basically the precipitation, okay? And in this precipitation step, you are separating out the proteins, and it's dealt with lowering the solubility of the reagent, means, or by adding certain reagent. Like if you recall in the DNA precipitation step, that is we use the ethanol, right? The absolute ethanol or isopropanol which is being used, and then in this ethanol we also added certain salts like NaCl or so. Why we are doing that? We are doing it because the solubility, or this is a polar molecule, the when you are using this, it is taking out the charges with the DNA because it's more polar as compared to the DNA. And second thing, when you are adding the ethanol, the DNA is not actually miscible, or you can say can dissolve. That's why it is nothing, whatever the residual water has been taken up by this ethanol, it is not miscible, then you precipitate out. The same phenomenon or the same principle is also used for protein isolation or the separation, that you precipitate by adding decreasing the solubility. And it is also associated with the three-dimensional structure. We'll see this in the next slide. Let's say for this in a picture, I've shown shown here. Now if you see in this particular picture, they're shown as a native protein. Okay, the native protein, if you look into the hydrophilic residues, will be on the outside because of the water, and the hydrophobic residues will be in the inside. That's how the three-dimensional or a coordinate structure of protein looks like. But when you add certain reagents, now these reagents, several reagents could be used like urea, guanidinium hydrochloride, glycerol, ammonium sulfate. Now there are two mechanisms in which they work out. Like for example, in ammonium sulfate, if you're talking about, if you add into very low quantity, then actually it increases the solubility of your proteins. That is called as opposite of this, which is called as salting in phenomenon. But when you increase the concentration of this ammonium sulfate to a more like 20%, 30%, 50%, 70%, then the solubility of your protein is decreasing, and the ammonium and you are basically precipitating out your protein. In case of urea, which is actually used as a denaturing agent, and they basically affect your hydration cell, that is which is shown over here. They flip the all the residues which is there, and they basically denature the protein. Now in some places, in fact, people can refold the protein, because if the protein is being defolded, means denatured, or its structure has been affected, so there are certain renaturation buffer people use. Sometimes I am taking this as a liberty to tell you this also as a knowledge point of view. Like when you are going for heterologous protein expression, sometimes your proteins is not coming as a soluble form, it is also making your aggregates, or which is called as inclusion bodies. Now to get rid of this inclusion bodies, sometimes you use this reagent in a low concentration like urea, and then you give into a refolding buffer, and then your proteins can come into a native structure. We will can talk about that in later point. But what the idea which I'm trying to say here is that the urea or guanidinium hydrochloride, they selectively partitions your hydration sphere. In fact, this guanidinium hydrochloride is also used for your RNA isolation. They have been guanidinium hydrochloride or guanidinium isothiocyanate, which is used to get rid of proteins as a protein contaminants. The ammonium sulfate, which is routinely used in laboratories for the precipitation, and as you increase, you selectively get the proteins of different sizes. Okay, so that's the salting out phenomenon, and what you are ultimately getting is your precipitation of the protein. Once you precipitate it, or you are preferentially extracted out your proteins from rest of the components, obviously for DNA and RNA, which is there, you basically get rid of, like you give the DNases or RNases treatment the same way you give the protease treatment, and we will be talk about that in our the protocol discussion. So that's how you basically precipitate. Once you precipitated your protein, now the thing is you already use a ammonium sulfate over here, or you have also used say urea or whatever things. How to do that? So that is another step which people use, that is called as the dialysis. You keep your protein, the precipitated one, in this dialysis bag, and this is a concentrated solution in buffer and outside. So after some times, let's say 4 hours or 6 hours, there will be an equilibrium, and then you have this protein, usually a cellulose bags, which is used for the dialysis purposes. Even there are certain columns which also use nowadays for this dialysis buffer. There are certain centricons, they are called as, which have a preferential pore sizes which can hold up to your proteins, and by using a centrifugal force one can also use, because dialysis sometimes take more time. Even for concentrate the protein, people also use that. Okay, the next part we will be talking about here is your column chromatography. Okay, so the column chromatography. Now this column chromatography, one of the widely used technique even for protein purifications. I guess some of you heard about it, which is called as HPLC, high performance liquid chromatography. It's essentially based on the same principle. So let's see how it's work upon. There is like other variations like FPLC is also there. Okay, and acta prime, which you will find in most of the national laboratories. So in this column shown here is you have two things. One is called the stationary phase, and one is called your mobile phase, or the buffer which you use. So as you can see here, there's a mobile phase has been shown, and to this matrix could be your agros. I think I have here, see, yeah, these are the materials. You can use a dextran, like just Cephadex column or Cephalus columns or a polyacrylamide column, whatever the size, pore size is different. Now, how this is working? First, you pack your column. Sometimes this is also ready-madely available with the different pore size, whatever the protein you have. You add the solution. Now, the how to isolate the solution, that's we will deal in part 2. Let's say you have the protein solution you have added, you load it into this column, and after loading into this column, what happens now? There is a large molecules, okay, like say this is a large molecules. Let's say I just make it like this, and there is a possibility of a small molecules. Now what happens, the small molecules which is there, or the size, they basically trap in between these pore sizes, and the large molecules like this, they basically elute out, and you collect these fractions. This is a very the principle is same. Now if you for a high performance liquid chromatography or high pressure liquid chromatography, you add the pressure, you make it the column very thin so that you get a very pure protein amount. That's why it is being used. You can use like what I was mentioning about the acta prime. They, it is a robotic ways you can actually use this, means there are certain fraction collections. Rather than doing it in manually, the robots actually collects based on your flow rate which is coming, and you have a different fractions which has been collected. This also nowadays is associated or connected with your UV detector and computer, so you can see where at what time, particular time, it your peak is coming. Okay, where is the more amount of protein? So that's how the column chromatography, and this chromatography is also called as gel filtration chromatography. So let me write here. So there is called as gel filtration chromatography based on the size. Some places you will find that it is also written at gel permeation chromatography, and both are actually comes in your size exclusion. That is a broad term which is used, that is size exclusion chromatography. Gel filtration is when you use an aqueous solution, means with the mobile phase, and if you use a organic solvent, then that becomes called as gel permeation. But essentially, it is based on the your size difference. Okay, so that is one way to separate out your protein. Now, this is, if you see in this particular the intensity, that amount of the protein which is coming. So let's say these are the peaks A and B. This is something which is a very small molecules, that is VT, means it will be retained somewhere in your column. Okay, somewhere in the columns they will be retained, and something which is a large molecules like this one will come out very early. So that is the basis of your gel filtration chromatography or gel permeation, and as well as HPLC, I would say. The next which people use here is your SDS page. The SDS page is basically you use a reagent which is called as the sodium dodecyl sulfate. Now this is a anionic detergent. Okay, and when you use this, and you also use a heat, okay, for doing this. So when you use this, you basically denaturing your proteins. The charge on all the proteins is basically will be you can say the negative charge, and based on the SDS, whatever the charge initially they present, then the migration which you are finding here over a gel is based on their sizes. So that's the SDS page. We will do a separate lecture for that how and to see. But what you can essentially see in this particular diagram, there's numbers or the labels is not important, but what is important is like you can see your bands like this. These bands can also be cutted out, like say for example, if this is say your desired band, you can collect and cut these bands, and then you can also purify your protein from there. So that is a SDS page, and there is another version which people also use is a native page when we don't add the SDS. Okay, so this is your 2D gel electrophoresis. Like this is from I taken from this paper. This is a link. Now this is about a yeast, because we were initially talking about these. So you can see these are some of the dots, and this is a two-dimensional your gel electrophoresis, and this obviously is radio labeled in this particular case. Now people have used a different chemistry nowadays, but what you can see in this, in a range of 16 kilodalton to 115 kilodalton, and based on its pH, okay, the pH which is I hope you know about the zwitterion and the pK values of the proteins. So based on this you will get these bands in your gel. With this I will stop here, but to summarize here, what essentially I have told, that what is the basic principle of isolation of the protein. I talk about that this principle is based on the salting out. Okay, so the salting out. The second thing which I talk about is the column chromatography. Now in this column chromatography, one thing which is involved like this, the ion exchange chromatography, the same principle, but you use a cation exchanger or an anion exchanger for the ion chromatography, or a affinity chromatography. In affinity chromatography people also use strepta avidin biotin, like you can conjugate your protein to the biotin, and then you can purify the protein, or you have a specific let's say HA tag with your protein, and then you can use a column which can preferentially bat with the HA tag protein, the fusion proteins, and rest of the things will be passed out with the buffer. So with this I will stop here. Thank you very much.